back to fatty acid synthesis. Ooh, look at that, right into the recording. I am, yes. Because this is the section, this exam 3 material is where inevitably everything slows down. Except me, no. But I am gonna try and introduce some stuff after spring break where you guys will be making up questions, where you guys will, be kind of doing more in-class stuff with each other, because it is challenging at this point to start keeping it all in your head. If you've been doing well so far, this is where we kick it up into like 9th gear or 10th gear, another random thing. So, I had a car before the one I have now, which was a 5-speed. It was a 5-speed automatic, but you could use the shifters. It had like a slapstick, so you could shift it in there. And I would actually improve my gas mileage by using the manual slapstick in there to change the gears. Well, the car I now have is an 8-speed, and that's X1. Yeah. And that is a lot different gearing. And the worst part is, for the Volkswagen, the gear shift was forward. For the BMW, it's backwards. So I have not trusted my muscle memory in there after like 14 years of riding in there. But anyway, we're jumping up gears here. So we had talked about the creation of fatty acids within the cell, how they tend to be cytosolic. And that's because we oxidize fatty acids in the mitochondria. So we want to separate those two processes so that we do not have a futile cycle where we're making fatty acids and immediately oxidizing them and just kind of burning energy, spinning our wheels for no reason, to continue the automobile metaphor. Woo. I will build analogies. That's my real skill, apparently. Anyway, but one thing we see is that we make a lot of acetyl-CoA in the mitochondria, and our fatty acids are made in the cytosol. So how do we get the acetyl-CoA to the cytosol where we can make our fatty acid synthesis? There's a transport system, technically two, because we got 2 membranes to cross, and it's going to cost us ATPs. So for every 2 carbon unit that we add, remember we have to spend an ATP unit to charge it to get it hooked together. We also need to spend 2 more to get our raw materials there. I can make jokes about supply lines and logistics and whatnot, but we'll try not to do that today. So we got to spend 2 ATPs to get our acetyl-CoA where it needs to go, and then a 3rd ATPA to prime it for the actual synthesis reaction from fatty acid synthase. All right, so let's take a look at this in terms of what's actually happening here. Where are we? So we got our matrix on the left-hand side, our cytosol on the right-hand side. We make our acetyl-CoA in the mitochondria, and then our citrate synthase is going to convert it to citrate. We're going to transport that out. Our citrate is going to get converted to oxaloacetate, and then our oxaloacetate to malate. But when we convert our citrate to oxaloacetate, we actually regenerate that acetyl-CoA. Okay. You see we're also burning some NADH in here as well, some NADPH if we go back through the pyruvate transporter or our aspartate malate transporter in here. And then, our pyruvate can be reacted to form malate and then oxaloacetate, and then work with our acetyl-CoA to get it in there. So we've got multiple places where we're spending ATP and our NADH is in here just to get this out. So what do you need to know from this? I'm not going to make you memorize this little cycle in here, but I do want you to know that this shuttle exists, that there needs to be a way to get acetyl-CoA from the mitochondria, where it's from the matrix where it's made, into the cytosol where it can be used for synthesis. Okay. All right. So we could even say fatty acid oxidation here leading to acetyl-CoA, but that's just one of the multiple ways we get acetyl-CoA in there. But we convert it to citrate to pump it out. We got 2 transporters, a citrate transporter in the inner mitochondrial membrane, and then a general pore in the outer membrane to allow it out there. And then we regenerate it. So it's just kind of the reverse reactions are happening on either side of the mitochondria. All right. So our acetyl-CoA carboxylase is our rate limiting step in there. Acetyl-CoA carboxylase is inhibited by palmitoyl-CoA. So if we make our palmitate, but it's still attached to a coenzyme A, that's our palmitoyl-CoA. We have this nice little 16 carbon CoA molecule. It will actually bind to our acetyl-CoA carboxylase and feed back and inhibit it. On the other hand, ACC is activated by citrate. So I guess we should give all like the basketball teams oranges, right? Oh, any mnemonic that will help. So if we have excess citrate, because remember our citrate, we're going to be converting into, acetyl-CoA in the cytosol as we transport it out there. So excess citrate is going to activate ACC in there. Okay. Basically, having high levels of citrate indicates excess energy, so we can store that long term as fat. Okay. And we see here again, that's just kind of going out that way. All right, so citrate is kind of a feed forward activation. Palmitoyl-CoA is a feedback inhibition. And then we have hormonal control, which we're not really talking about too much yet, but it's coming up in chapter 23. That's where we tie everything together in here. But we'll see things like glucagon and epinephrine are going to inactivate acetyl-CoA carboxylase. Glucagon says, I'm hungry, so we're going to stop making fatty acids and start cleaving them. Hopefully, says Dr. Ebert. Epinephrine is like, need energy now. So we saw epinephrine, release glucose. We're going to, stop making fatty acids. We're going to start breaking them down. And both of those act through a phosphorylation cascade. They bind to their receptors. We're going to see a bunch of phosphorylation go on. We're going to phosphorylate ACC, and that's going to prevent it from making our product, right? Our malonyl CoA. Okay. So to go into a little more detail on that, remember phosphorylation is covalent modification. We're actually attaching a group to it. So in this case, it's a phosphate group. That's probably our most common covalent modification within enzymes for controlling there. Basically this phosphorylation, the more ACC is phosphorylated, the less active it is. So it's active as dephosphorylated monomers. When we dephosphorylate it, it polymerizes, and that's where we go to this picture up here, right? I think this is our, let me check to make sure I'm telling you the right thing on this. Yes, so that little electron micrograph on the far right is filaments of active acetyl-CoA carboxylase in there, right? So we actually get these nice little filaments when it's dephosphorylated, because these long things can kind of come together. When we phosphorylate it, it breaks those apart. So we just have little pieces out there. So it's kind of like you can think of it as a self-assembling shirt. That's terrible. It's terrible. But when it's dephosphorylated, it all comes together into its active form. You can wear the shirt. When you phosphorylate it, it all falls apart into a pile of thread, which is not active, not useful. Not active wear. Any mnemonic that works. Right? Okay. There are also other ways to regulate fatty acid synthesis. So we've got feed forward activation, feedback inhibition, covalent modification. We also will see gene expression. So if you change your diet, yes, I'm trying so hard. I've not had red meat in like a week, aside from a few pieces of beef jerky for my lunches on Tuesdays and Thursdays, but we're gonna pretend that doesn't count. We can change gene expression in there. We can look at various transcription factors. These specific transcription factors that affect the gene expression for fatty acids and their enzymes are called peroxisome proliferator activated receptors, or what is much more fun to say, PPARs, right? It's just fun to say. And, I think of that in the springtime because you got the little frogs that come out there that are like PPARs. I don't know. It's, the plants are starting to grow. Yeah, the plants are starting to grow. If we did plant, we could look at plant fatty acid synthesis. So that's weak. That's a terrible one. I don't like that one. We also have reciprocal regulation as well too. So for example, if we have a lot of malonyl CoA, we're going to inhibit fatty acid transport into the mitochondria. Okay, so that basically means we're not going to be sending fatty acids to be oxidized, which means more fatty acids as they're made are going to accumulate in the cytosol. So we'll end up making more of that palmitoyl-CoA, which will feed back into ACC. Okay. All right. But what this malonyl-CoA, by binding to the, by inhibiting the fatty acid import into the mitochondria, that is just another way to help. Besides separating those two locations physically, it just makes sure we're not shuttling fatty acids back in as soon as we make them, that we're doing something else with them, sending them for storage, usually in the adipocytes. Okay. All right, so hopefully that's not too bad. All right. Yeah. All right, cool. So we'll switch gears ever so slightly here, because apparently I have a car themed lecture today. We only make palmitate. We make a 16 carbon saturated fatty acid, and that's it. You get one product. Here's your oatmeal that you're going to have for breakfast every day for all year. But maybe we want some variety. All right. So what can we do once we've made the palmitate? Okay. There are ways to lengthen palmitate to longer chain fatty acids. Okay. So these elongation systems are found in the ER. And some in the mitochondria. It's like, wait a minute, didn't we just say we don't send the fatty acids? Yeah, everything's a lie. But we're going to focus mainly on the ER version of this, and we're not going to focus on it very much at all. But the key is, just like palmitate synthesis, we're going to add 2 carbons at a time. And the most common product is stearate. Hey, we're just going to add 2 more carbons. We go from 16 to 18. Okay. But we can also desaturate these fatty acids as well too. So we can, introduce a double bond in there. So our palmitate can become palmitoleate, or our stearate can become oleate, where we introduce a double bond at carbon 9 in that fatty acid chain in there. There are desaturases that exist in animals. These are also, fatty acyl-CoA desaturase is the longer name, but we'll often just call them fatty acid desaturases. These require NADPH, so we're getting our reducing power from NADPH instead of NADH in this time. And we're going to use a couple cytochrome B5 and B5 reductase are unusual ones that other than this, I've never talked about them. We can specifically call this a delta 9 desaturase because it reduces the bond between carbon 9 and carbon 10. So theoretically, you could have like a delta 11 desaturase or a delta 5 desaturase, or I don't know if we could have a delta 6, but anyway, we'll see. How does this work? Well, just like we saw with the electron transport chain, we got a whole lot of electrons moving through a cycle here. So here we get our NADPH, donates electrons to the reductase, to the cytochrome B5, and then this is going to trigger our power here to give us the reducing power. And we'll basically have this elimination of water. So this is a dehydration reaction to make this alkene. All right. 2 of the electrons come from the saturated fatty acid, and 2 of them from our NADPH ultimately, directly from the ferrous cytochrome B5. Okie dokie. So we can do delta 9 desaturases, but plants, well, actually, and we can also do 4, 5, and 6. So we can do that there. Plants can actually have multiple desaturases in here. They can go on and do more. They can, basically have a double desaturation, delta 9, delta 12, delta 9, delta 12, delta 15. So what this means is that we can't make all the fatty acids we need. So we have to get them from our diet. So we got to go chomp down on some plants that make these. And these are our polyunsaturated fatty acids. They're critical for controlling membrane fluidity. All right, so we want to introduce those cis double bonds in there so we can make a more jumbled membrane so that it doesn't. Yeah. But these are our PUFAs. So today's lecture is all about PPARs and PUFAs. We also will utilize these polyunsaturated fatty acids as precursors to making eicosanoids, more biologically active lipid-based biomolecules, which we'll talk about here soon. Eicosanoids. So if we look at things like, I think they're precursors to like the clotting factors in your bloodstream. So if you want to make thrombin or leukotrienes, but we'll get to the eicosanoids here soon. It's all coming. It's just, it all ties together. Makes me think of like some TV shows. So you ever have it? You know, most TV shows, when you watch them, they have a beginning, a middle, and an end. But every now and then you'll get one where there's like a bunch of people involved, and then maybe you'll see like all the things that happen to one person, all the things that happen to another person. So it's not chronological. Our chapters are not chronological because we're touching so many things are interrelated. We're saying, yeah, we're gonna make these eicosanoids. More on that in a moment, you know. It'd be like, you know, reading a book where it says, see page 48, see page 52, see page. So that's the tough part here. All right, our plant desaturases, I'm not going to talk about these too much, other than to say they're a little bit different because they will act on fatty acids that are attached to a phospholipid. So here we have, you know, a nice little phospholipid, and these saturases can actually, you know, alter the composition of the phospholipid. Apparently, it's time for my stomach to growl. So, yeah. All right. So how does all this happen? All right. So we can look at this in general as saying, well, first thing we're going to do, we're going to convert pyruvate to oxaloacetate. There's our pyruvate carboxylase that we talked about. Pepsi K is going to give us phosphatidyl pyruvate. We're going to have a whole bunch of steps to give us dihydroxyacetone phosphate. We're talking about gluconeogenesis, right? Look familiar? Yeah, so we get to dihydroxyacetone phosphate, and instead of going back to make glucose, we're going to shunt that over into glycerol. All right, so remember, glycerol is our 3-carbon subunit, and we're going to make glycerol 3-phosphate dehydrogenase, and that is going to be our base molecule for making triacylglycerols. All right, so now that we have this G3P, we can attach fatty acids to carbon 2 and carbon 3 and make our tags from that to make our phospholipids. Okay. Or we can get rid of the phosphate group and make a triacylglycerol there. All right. Apparently, I really need the bigger breakfast. All right. Eicosanoids are short-range hormones made from arachidonic, arachidonate, or arachidonic acid, which we get from spiders. You guys know you eat like a couple spiders every year. As you sleep, a spider will crawl in your mouth and you'll chew it up and eat it. Yeah. And I always like to tell people that's where you get arachidonic acid from, but no.
That's like the true thing. That's not the same.
You eat a certain number of spiders every year without realizing it. Yeah.
They want to live inside of our
Yes. Yeah.
Don't do that.
I haven't seen spiders in the house.
Yeah. But our eicosanoid hormones are our prostaglandins. That's the one I forgot. Leukotrienes and thromboxanes. All right. So arachidonic acid gets incorporated into the phospholipids of the membranes, and then there is a particular phospholipase that will cleave those off and release the arachidonate, and then it can be used as a precursor for all this. So you can see here we have this enzyme is called phospholipase A2, right? So that tells us that maybe there's an A1 or an A3, which would cleave any fatty acid groups at position 1 or position 3. Usually not 1, because that's going to, if this is a phospholipid, that's going to be where the phosphate group is. Usually not A3, because that's usually a saturated fatty acid, and we're more interested in this, so we're going to call it A2. More likely, we're going to see phospholipase B, C, D, or something like that has a similar action. And the 2 is just telling us where it acts at. All right. So we're attacking our carbon 2, releasing this unsaturated arachidonic acid for use later on. So arachidonic acid is a 20-carbon acid. It's got 4 double bonds in it. And if we go back to chapter 10, we draw it as a nice straight line. Well, we're going to fold it in half because we're going to do stuff with it here. And when we fold it in half, we can kind of see where we're going here. All right. Our enzyme, we're going to have this cyclooxygenase activity, and that cyclooxygenase enzymes, we're going to call like COX-1, COX-2. Those are the two biggies. We see these in inflammation, right? So one of the things that happens if you get injured, you will see inflammation, swelling that'll happen. A lot of times to avoid that and to avoid fever, we can use various anti-inflammatory agents like aspirin or ibuprofen or naproxen, right? So they will inhibit this cyclooxygenase activity, okay? But assuming that we just go forward with this, our cyclooxygenase, you can see here is our cyclo and our oxygen form. We're essentially creating, it's not really an epoxy, but it's a ring structure that's got two oxygens attached into it there. We'll also see we'll have this peroxidase activity because here we have this peroxide subunit. We're going to just convert that into a hydroxyl group. So basically we have our PGG2, this prostaglandin form here. The peroxidase activity will give us PGH2, and then we can continue the reactions here to make other prostaglandins. We can also start converting these into thromboxanes as well too. But our initial step for all these icosanoid class of let's say proteins, but lipids, signaling molecules, bioactive molecules, we take arachidonic acid, fold it in half, and introduce this cyclooxygenase activity in here and this peroxisome in here, right? So we're basically adding a couple oxygens to make a PGG2. We're basically adding two O2. So here's the first O2 and here's the second O2. Then we remove a single oxygen to give us the group in here. So basically what I want you to know, we cleave off arachidonic acid from a phospholipid, and then cyclooxygenases act on it to make our prostaglandins, and then those can continue down a variety of pathways to make other icosanoids, okay? So if we look specifically at PGH2 synthase has two isoforms, COX-1, all right, and COX-2, all right, okay. COX-1 is going to catalyze primarily prostaglandins that regulate gastric mucin secretion. That sounds lovely, right? So all the slime in your gut and your stomach, the stuff that helps, food absorb and kind of protect the villi a little bit. You get lots of turnover in there, but so you're not kind of dissolving the proteins in your that make up the lining of your stomach. Is this mucin is important in there. Yeah. So all the lovely slimy stuff in there. COX-1 does that. COX-2 catalyzes the synthesis of prostaglandins that mediate pain, inflammation, and fever, right? So what's one of the big problems with aspirin? You take a whole bunch of aspirin.
You said, slowly break it down.
Well, no, I don't mean a whole lot at one time, but I mean if a lot of people can't tolerate aspirin very well because they'll get an upset stomach, and if you take I don't want to say high doses, but constant. If you constantly take aspirin or similar NSAIDs like that, there's a risk for developing ulcers in the stomach. Why? Well, this aspirin non-specifically affects all the cytooxygenase enzymes in there. So we want to stop COX-2 because we want to reduce pain, reduce inflammation, reduce fever, but this one, not so much, all right? So if you look at Bayer, why did Bayer become a giant company in Germany? They figured out, well, actually, I think they were involved with fertilizer maybe, but anyway, with medicine, one of the things that I think they were one of the first companies to come out with buffered aspirin, so that the aspirin wouldn't go directly into the stomach, but the stomach would slowly dissolve the coating around the aspirin, and then it would be absorbed primarily in the intestines to try and reduce any stomach upset that you'd get from that. All right. So if you were to go find willow bark, and this is actually how aspirin kind of came about, people noticed that if they were injured or sick, if they chewed on willow bark, they'd feel better. You can still find people that will make like willow bark tea. Willow bark is high in salicylic acid. Salicylic acid is the precursor to aspirin. That may have been something you did in high school chemistry class because it's actually pretty easy to make aspirin. I mean, we've been making aspirin for chemically 150 years, maybe quite a long time now. But it's an easy thing to do. Things I can't recommend, but why I definitely ended up in science. In my high school chemistry class where we made aspirin, we were supposed to make it. And yeah, there's an acidic residue, so you got to wash it really well to get rid of all the excess acid. I was super careful, and we're supposed to throw our aspirin away, but not me. I waited till I got a headache. And then, because it was a long time ago when, you could have razor blades in school, I cut mine up with a razor blade and snorted it when I had a headache, and it got rid of the headache. But, for real. Yeah. But I was obscenely careful with how I made my aspirin.
No, I just trying to make sure.
Yeah. And that's one of the crazy things. Yeah. I mean, that's one of the things about being a developed nation. And actually, and so that made me think, because I'd never heard of Goodies powder at that point. But the Goodies powders were, why do Goodies powders act so fast? Because you're snorting them. Well, they go directly into the mucous membrane, so they get absorbed a lot faster than if they have to go into the stomach. So it's a faster acting pain relief in there.
No.
You can mix them in yeah.
Doing these wrong.
Yeah. So,
like you practicing for a line of code.
exactly. That's what we all pretended to do in high school because we were dorks, and that was before cocaine actually made it to our high school.
Question: around what era were you in high school?
Oh, let's say the late 80s.
Oh yeah, that was during the crack era.
Yeah. Actually, I had friends who spent time. I don't know if it was the late 80s or early 90s, but in Miami, it was dangerous. That's when the whole Miami Vice thing was there too. But there's a good reason why Miami Vice was a show, because Miami had a lot of seedy areas, and he actually saw people get, well, maybe killed, but stabbed and seriously injured. He's like, yeah, maybe I don't want to be in Miami that much. But anyway, back to this cyclooxygenases. There's two of them. One that controls stomach mucin, and one that controls prostaglandin synthesis that affects pain, inflammation, and fever. All right. So our various NSAIDs are aspirin in there. Aspirin is an irreversible inhibitor, so it will bind and just kind of take that out. We get a lot of turnover in our cyclooxygenases, so it kind of takes out the ones that are active. So we make some that are fewer in there, and you can see our salicylic acid to aspirin in there, acetylsalicylate. Ibuprofen and naproxen, on the other hand, are competitive inhibitors. They resemble the substrate. All right. So if you go back and kind of look at that folded over arachidonic acid, you can say, well, that sort of looks like that. But they resembled enough to bind in the binding site and inhibit the activity of cyclooxygenase. Yeah, but aspirin tends to have a higher stomach upset than these two, probably because these are reversible, and this, is irreversible in there. So we actually end up transferring that acetyl group to a serine in there, which inactivates that. Okay. Is this where the drug thing is? Maybe I'm getting ahead of myself. All right. We may talk about this later, but in the 90s, there was a new drug that was developed that only inhibited COX-2. So no stomach upset. All right. And what was the name of this drug? It's actually back on the market now, but I want to say it's Celebrex or Celexa or yeah, I can't remember. But it was a drug designed specifically to treat pain. It might be Celebrex. Yeah.
Celexa is
Was it antidepressant? Okay, yeah. Yeah, I should ask the farm tech. Yeah. But when it was first marketed, it was awesome. And the pharmaceutical company was like, we're gonna make millions. attack and stroke. Significantly enough that the FDA pulled it and put a black box on it. And now there's, I'm not sure. I'll have to look at it. It might be coming up later up in my slides.
Yeah, because that is one of the COX-2 inhibitors. And this is very popular.
Yeah. And, it's prescribed for a lot of older people. It's something they could take daily because, arthritis, aches and pains of aging and whatnot. So, yeah. Meanwhile, I've started doing radio Tyson. Oh, Lord. So those are our prostaglandins. We can also look at leukotrienes, all right? This also starts with arachidonic acid. We have lipoxygenases as well in here that can, take our arachidonic acid and we start making, we're just going to call this 12-HPEAT because hydroperoxy icosaptetraenoate is a mouthful, but we'll call it 12-HPEAT. That's our initial one for one class of leukotrienes. We also have 5-HPEAT, and it just depends on where we're adding this peroxide group. Okay. So these lipoxygenases are mixed function oxidases. We're basically adding peroxides. We're adding oxygen directly to the arachidonic acid, and we can make a series of leukotrienes. We're not going to go into what all they do, but these are, again, critical. And just like our prostaglandins, these are critically important in here. So it all stems from arachidonic acid. So we need to make sure we have enough arachidonic acid and that our pathway is moving forward here. All right. I wonder if I should talk about the thrombins as well too. I mean, blood clotting and whatnot. I think we mentioned thrombins and blood clotting in the previous chapter somewhere, but thromboxanes again come from this pathway. So arachidonic acid, critical dietary consumption. All right, if we look at our triacylglycerols and phospholipids in animals, plants, and bacteria, then we get into another little topic area here. Okay, so where do we get these from? Seeds and nuts. It's like Dr. Ebert's morning breakfast. It's like nuts and berries now. I feel like I'm a foraging animal, right? If we look at the average 70-kilogram human, so that is what, about 154 pounds or so, you've got about 33 pounds of fat. So roughly 20% of body weight is fat typically. This is enough to survive for 12 weeks. So if you get lost in the woods in the wintertime, you don't freeze to death and you have enough water, you could survive for 3 months, whole season in there. So our fat stores, we measure in weeks. Our glycogen, we're looking at hours. All right, so maybe we got half a day as opposed to whatever glycogen. And they say in muscle, but our muscle glycogen is kind of weird because it's hard to use up your muscle glycogen, but there's not very much of it. Animals, plants, bacteria make phospholipids for the cell membranes. All those molecules contain a glycerol backbone and two fatty acids or 3 fatty acids, depending on whether we're talking about phospholipids or tags, respectively. I'm just going to call it triacylglycerol tags, TAG, for the rest of the thing, because that's a lot easier to say. And, that could be awesome graffiti, right? You could come up with your own personal fatty acid and then tag various buildings. Okay, sorry. Yeah, whatever. Any mnemonic device that works. All right. So where do we synthesize these? We've made our fatty acids, but remember, we can't really transport fatty acids too well. So we're going to assemble them into either phospholipids or triacylglycerides. All right. So in order to do that, we need our glycerol. And we saw, we basically, if we go through gluconeogenesis, we make that dihydroxyacetone phosphate. And then rather than continuing the glucose, we're just going to shift that, shunt that into a new pathway to make our glycerol 3-phosphate. Okay. Yeah, I should probably switch that previous slide around with this. This is made via something called glycerol kinase, and it's a minor pathway found in the liver and the kidneys. Okay. So we're making our fat in the liver and the kidneys. That's why you may hear about diagnoses of people who have like fatty liver disease. Make triacylglycerols in there, and then maybe can't transport them out or something. That could be one of the causes. All right. So what we do first is we make phosphatidic acid. That's our first component, and we make that before we make other triacylglycerols. Right. So the thing about this is what we're essentially doing is taking this phosphorylated glycerol and attaching two fatty acids to it, carbon 1, carbon 2. Okay. All right. And you can see here's our acetyl-CoA synthetase, attaching those on there. Here we have our two R groups, our fatty acid chains. And what we get is a phosphatidic acid. All right. So there's an advantage to doing this because now we have a phosphorylated glycerol group with two fatty acids. We can leave that as a phospholipid, attach something else to that phosphate group and incorporate it into the membrane, or we can get rid of the phosphate group and attach a third fatty acid to make a triacylglycerol and then transport that to the adipocytes. Okay, so that gives us a bigger pool of choice as we start from there. Okay, so here's our phosphatidic acid. Again, we can kind of cleave off the phosphate group, leave behind a hydroxyl group. So we have a 1,2-diacylglycerol. I think we sort of saw this in a pathway that it didn't emphasize too much with hormone-sensitive lipase in one of the earlier chapters in there. But we'll have an acyltransferase that can add another fatty acid group to make our triacylglycerol, or we can just attach something directly to this phosphate and get a glycerophospholipid. So there's two pathways. And you can see often this isn't as simple as it looks because we have multiple arrows here to attach various stuff. All right. So in this part of the chapter, we're going to start talking about how we regulate specifically triacylglycerol synthesis. And we're going to look first on insulin. Okay. So, and this is where we get into, oh, I can't say that because I got to get the exams back. Yeah. Well, I still got some other exams to, yeah. But yeah. Put you guys in your own room now. I'm gonna unrecord this part now. Just kidding. But insulin by itself is going to stimulate triacylglycerol synthesis. Why? Well, when insulin is released, it's released in response to glucose in the bloodstream, which diet. So if we have elevated levels of glucose, we're like, oh, good, we got lots of energy. Let's burn this glucose that we have for energy. We'll store some more glucose for later. But since we have so much energy, we might as well make some fatty acids for much later, right? Okay. What we see here, insulin upregulates our acetyl-CoA carboxylase so that we can make more fatty acids and triacylglycerols. However, if we don't have insulin, right, then we'll see increased lipolysis, where we'll start cleaving these down, increased fatty acid oxidation. And if we, you can get to a stunning amount of ketone bodies being made in here that will basically have our oxaloacetate that gets depleted in there. So that's where you can get the diabetic coma, all the good stuff in there. And if we don't have insulin, we're not going to make fatty acids. We're just going to be oxidizing them. We'll talk about keto diets and whatever, all that good stuff in there. Triacylglycerol breakdown and resynthesis create a futile cycle. So for much of this course, I've talked about, oh, we want to separate these two processes. I just talked about fatty acid oxidation in the mitochondria, fatty acid synthesis in the cytosol, so we avoid a futile cycle. Well, you go into the adipocyte, and we have a futile cycle. 75% of the free fatty acids that are released by lipolysis in the adipocytes are immediately reesterified to make triacylglycerols rather than used for fuel. So we basically just kind of cut off a piece. It's like, do I need it? Nope, not yet. Put it back on. Cut off a piece. So even when you start to go through, low energy states or withdrawal or early starvation, this is happening. Even in late starvation, you're still reesterifying. these as you make them. Maybe not quite at the same rate, and you start to lose some of those fat stores in there, but that happens. Some recycling in the adipose tissue, some are transported to the liver, remade into triacylglycerols and back, and then transported back to the adipocytes. The distribution between these two paths will vary, but again, we're going to look at about 3/4 of them are just constantly recycled in here. In low energy states, the one that gets transported out are probably used and not sent back. So you see a higher and higher increase in the reesterification within the adipocyte cells themselves as you go into low energy mode. But overall, even in starvation, you're constantly reesterifying these. All right, we have a tag cycle. All right, so of our adipose tissue, we can generate our glycerol 3-phosphate in the adipose, make a triacylglycerol. We can cleave off those free fatty acids, and we have our glycerol there too. Our free fatty acids can get transported through the bloodstream. They can be sent to various tissues as fuel, or they can go to the liver and get converted back into triacylglycerols, which can be released into the bloodstream, for fuel, or sent back to the adipose tissue for storage. They cross the adipose tissue as free fatty acids, and then get esterified into our tags for storage in there. Okay. You can also see we can use glycerol much in the same way that our glucose. Remember, we break that down to DHAP and G3P, so we can continue down that pathway. Glycerol can feed back into that pathway for glycolysis as well. We're going to see a big role of lipoprotein lipases as well too, which we'll get to here in a bit. Okay. I think I've actually said this like twice before here. I really need to clean up these slides. They're kind of messy slides. These are not as organized as they should be. But our glycerol 3-phosphate, that is critical for tag synthesis or phospholipid synthesis, comes from DHAP from gluconeogenesis. All right. So we can really call this glyceroneogenesis as well too. Just instead of making glucose, we just take a little side road to make our glycerol instead. So during lipolysis, which can be stimulated by either glucagon or epinephrine, glycolysis is inhibited, so we don't have DHAP readily available to make our glycerol 3-phosphate, and the adipose cells won't have glycerol kinase to make them on site. So glyceroneogenesis will basically contain some of the same steps of gluconeogenesis. Basically, we're just converting pyruvate to DHAP, and so it's abbreviated gluconeogenesis. And here's the same picture again. I really do need to go through and clean these up. I think that's probably making the slides over a series of several days. All right. So we make our triacylglycerols, all right, and then we can store them in the adipocytes, or we can transport them to where they're needed as free fatty acids and use them for energy. Okay. What about our membrane phospholipids? The other half of that pathway, where we have our phosphatidic acid. We still have we've added 2 fatty acid groups to our glycerol phosphate. Now we have the phosphate head group, and we can put something else on there. All right. We're essentially, whether it's a phosphatidic acid or a diacylglycerol, we can do this, go down this pathway. And there's typos in here. Oh, that's terrible. Okay. So if we want to attach a phospholipid head group, we're going to use CDP, our cytidine diphosphate. All right. So, whoa, we talked about using ATP. Everybody uses ATP all the time. GTP, we've talked about that a lot. Well, here we have CDP, cytidine diphosphate. So, we attach cytidine diphosphate to that head group in there. Okay. We're going to we're going to release cytidine monophosphate. All right. And now we leave behind this glycerophospholipid with the head group attached to the phosphate there. So our head group basically attacks the second phosphate group and cleaves off the CMP. Our second strategy is we could have our CDP directly bind to the head group, and then the alcohol attack that second phosphate and again cleave off the CMP. So it doesn't really matter whether we attach CDP to the phosphatidic acid or to the head group that we want to attach to make that. In either case, it works because we have an alcohol group attacking the phosphate group. Okay. So come from the left. Got to make it an interpretive dance, right? Okay. All right. The key is we have cytidine. Cytidine diphosphate in there. All right. If we look at E. coli, starting bacteria, I always like to compare bacteria to eukaryotes. So there are two main pathways. Phosphatidylserine is synthesized and can be decarboxylated to phosphatidylethanolamine. So we can see this. We're going to basically, add a serine group in here to our CDP diacylglycerol. So now we have phosphatidylserine. I know this is not super easy to see, but we can decarboxylate this and make our phosphatidylethynolamine. Okay. The other way we can go is we can add this. What are we making here? Phosphatidyl 3-phosphate group in here. So we have basically added glycerol 3-phosphate, and then we can start modifying that on the chain there. So we get phosphatidylglycerol, and if we add some more stuff in there, we can get this cardiolipin and so on and so forth in there. It's weird to think of bacteria having cardiolipin because they don't have hearts, right? But but we basically have two main pathways of phospholipid synthesis in E. coli. All right. Eukaryotes are similar, just a little more complex. All right. So we can look at again attaching phosphatidylglycerol in here through our CMP, cleaving that off, and ultimately get to cardiolipin. There's a replacement of CMP rather than glycerol within eukaryotes as opposed to bacteria. And then again, we can attach inositol here, releasing our CMP, and then go to make our phosphatidylinositol. And then we can phosphorylate groups on there and continue on making modifications. So the whole point of this is that CDP is our intermediate. We're going to release CMP, whether pretty much any organism we're talking about. Strategies are similar, but not quite the same. Okay. If we look at yeast can make phosphatidylcholine from phosphatidylethanolamine. Yeah. Oh. Yeah. Yep. Okay. Yeah. Yep. No worries. No worries. I'm not gonna emphasize this. I just want you to know that there are various enzymes and strategies across organisms that are going to start to make these phospholipids and attach various head groups on there. Right. All right. Yeah. And then it's kind of weird because in mammals, we've got a salvage pathway here because we go backwards from phosphatidylethanolamine or phosphatidylcholine. We just kind of do these exchange reactions, but we're not going to sweat that. Okay. I don't even want to. I should cut that slide because I don't want to talk about salvage pathways because those come up later where we're going to talk about real salvage pathways when we break down our. I think it's in the next chapter. All right. So basically our phospholipid biosynthesis, we could look at all this in here. The only thing I want you to be able to do is look at this pathway and say, oh, okay, if I want to make, phosphatidylserine, I can start with, a phosphatidic acid, of some type, and I can go to, this CDP diacylglycerol and then put a serine in there to release the CMP to give me phosphatidylserine. Okay. And then, so on and so forth. So just being able to follow this pathway is enough. Not memorize it. I think, yeah. Because this would be something that I would give you on a take-home questions. Like, what happens if, like some of those questions you got. All right. Now, the reason I actually do cover this is because we have a whole bunch of other things that are important that get derived in similar ways. So ether lipids and plasmalogens. So if we start talking about, I'm bumping up against my limit here. If we start talking about making our plasmalogens, which are important regulatory molecules, we have similar pathways in here. All right. So Same way, making sphingolipids. If you recall sphingosine, synthesis of sphingolipids are going to use very similar pathways. All right. All right. So that's all I want to say about those. More emphasis is what I'm going to place on this part coming up here. Let me check my time frame here. We're good. Okay. Sorry, I looked at my. Anyway, cholesterol synthesis is the second big thing we're looking here. So fatty acid synthesis in general, and now cholesterol synthesis and transport. Not yet, but well, we are going to make some steroid hormones here, but we're going to get to the cholesterol first and the steroids. All of these are going to come from an isoprene unit. So we have this nice little 5-carbon unit here. But our cholesterols and our steroids and our isoprenes are related but distinct from our tags, phospholipids, sphingolipids, plasmalogens. So you can think of those previous ones as like being glycerol based or sphingosine based, but they're over there in their class, our traditional 3 things attached to a glycerol backbone or 2 things attached to a sphingosine backbone. Our cholesterol, we're going to be looking at this ring structure that we come here. And we're going to see this is going to be built up using 5-carbon isoprene subunits. All right. So we're switching to a new box of Legos, in other words. Okay. So how do we do this? How do we make cholesterol? All right. Here it is in one slide. And actually, that's one of the things, as opposed to 40 or 50 slides that talk about fatty acid synthesis, one of the supplemental things I have on the exam in there is like fatty acid synthesis in 8 slides or 6 slides or something. So there's a shortened version of this on the campus modules. Cholesterol biosynthesis, this is the short version right here. We're going to take 3 acetates. We're like, oh, 2 carbon units are acetyl-CoA. Awesome. We're going to take 3 of them and make mevalonate. Okay, so look at 1, 2, 3, 4, 5, 6. We got a 6-carbon unit, mevalonate. Okay, awesome. We're going to lose a carbon. I think we make carbon dioxide. And we're going to condense that into a 5-carbon isoprene. Okay, so now we've got our isoprenes in here, and you can see we phosphorylated them. Must be some other steps in here, but these are activated isoprenes. Once we have our activated isoprenes, we can take 6 of them and mash them together to give us squalene. All right, so now we've got a 30-carbon squalene. Okay, and then we're finally going to cyclize that, so you can see if we kind of bend this around. Oh, we can make ring structures in here, right? Just like we see in cholesterol, and that's how we make our cholesterol. So we can kind of divide all this up into 4 major steps. Acetates to make mevalonate, mevalonate to make isoprenes, isoprenes to make squalene, and squish our squalene together to make our ring structures and our cholesterol. Okay. Good times, right? Yeah. So there's a lot to this, and this is important stuff to know. So because cholesterol is such an important molecule in the body. So how do we make mevalonate from acetyl-CoA? All right, we take our acetyl-CoA and add, combine it with it was 2 acetyl-CoAs here. Yeah, okay, 2 acetyl-CoAs. We combine them together and we get acetoacetyl-CoA. We basically take the acetyl group off of one CoA and add it to the acetyl group on the other CoA. So we release a free CoA. Okay, you see here, we now have this acetyl-CoA. All right, this is done by something called thiolase. But remember, we need to add 3 of these 2 carbon units. So our third is going to be attached on here by an enzyme called HMG-CoA synthase. All right, this is our hydroxymethylglutarol-CoA that we're making here. Okay, and the reason this is different because we're adding our carbon to one of the interior carbons in here, so that we don't have a linear carbon structure with our mevalonate. We have these 6 carbons with a methyl group coming off in the middle. Okay, and that's our HMG-CoA. Okay, and then our HMG-CoA reductase is going to remove that coenzyme A and give us mevalonate. Okay, so hydroxymethylglutarol-CoA gets reduced to mevalonate, and then we see our 1, 2, 3, 4, 5 carbons, and our 6th carbon is a methyl group off of carbon 3. Okay. So HMG-CoA reductase, we will end up looking at in some detail for regulation, because it is a target of many cholesterol lowering drugs, especially like the statins. So we're going to say, all right, you can get to this step, but nope, no more. We're not going to make mevalonate so that we can't, convert that into activated isoprenes to squalene to cholesterol. Right? So HMG-CoA is kind of one of our early steps of cholesterol synthesis, which is why it's so important. Okay. All right. We'll look at the regulation of that soon. Probably not today, probably after spring break. Maybe not that soon, I guess. Mevalonate then gets converted into activated isoprenes. Okay. All right, through a whole series of steps here, and you can see we're spending ATP in a bunch of these steps, and we do release carbon dioxide at one of these steps as well too. So we're going to cleave off one of those end carboxyl groups in there, and we can make these isopentyl pyrophosphates or dimethyl allyl pyrophosphates. These are interchangeable, but they are activated isoprene subunits. Okay. So do you need to know all these steps? No. What you need to know is mevalonate gets converted into these activated isoprenes, and it requires ATPs to do that. There's a whole bunch of steps. Okay. So no need to worry about any of those. Okay. Then we're going to take 6 of those isoprene units and condense them to make our 30 carbon squalene. I always, geranyl pyrophosphate. Oh, another typo. Jeez, that's terrible. Pyrophosphate. Yeah, it smells like shate to me. But I always, I know I missed the typo, because I always look at geranyl phosphate and think geraniums. It's like we're folding, you do origami and you end up with a flower and we make a geranium here. But yeah, yeah, not my favorite hobby either. But there's all these steps in here, but the key is 6 isoprene units to make 1 squalene. Okay, we're not going to sweat that. Again, it requires some reducing power in here, so we're putting some energy in here to make this, but we end up making squalene. Right. All right. So a bunch of this would get geranyl phosphate, 2 geranyl phosphates to give us we had a farnesyl phosphate. Doesn't matter. 6 squalenes, 6 isoprenes, 1 squalene. All right, and then we're going to make those ring structures in the squalene. We got some more origami to do here, and it's funny because we end up with an epoxide intermediate here, the squalene 2,3 epoxide. So epoxides are not very stable; they're highly reactive. We also see pathway divergent in animals versus plants. So in animals, we get lanisterol, which then gets converted to cholesterol. In plants, the epoxide is going to cyclize to things like ergosterol or brasinolide or something like that. Okay, so we're not going to worry about plants, but here's the deal. We have this epoxide down here at the end that is highly reactive, but that is going to allow us to, make these various compounds in here. So again, plants and fungi, you'll see, stigmasterol, ergosterol. We're going to focus on the central one, where we're going to make lanisterol, and then we kind of condense over here and lose these methyl groups to make our cholesterol. What do you really need to know? Squalene goes through an epoxide intermediate to make cholesterol or other sterols in other organisms. We're going to be humanocentric, anthrocentric. Mm-hmm. Almost certainly. I'll have to look at that right after. I'll look at that right after class. Okay. So big, huge, long pathway with a whole bunch of crap coming together to make cholesterol and a lot of energy put in there. So what happens to cholesterol after we synthesize it? So most of it's synthesized in the liver and then exported to other places in the body where it's needed. They can be exported as bile acids, biliary cholesterol, which is similar to bile acids, or as cholesterol esters, where we take that hydroxyl group at the end and esterify it in there. Bile is stored in the gallbladder and secreted into the small intestine after a fatty meal. So if you go have like a big steak, what's the cut of steak, like ribeye? Is it not? Is it ribeye? Prime rib. That's I like ribeye. I don't like prime rib because prime rib has so much fat in it. Yeah. And that's the thing. When you look at steaks, cuts of steaks, the marbling, the amount of fat in there to give it the juiciness. I'm a lean cut type of person, but anyway. But if you eat a big fatty meal, your gallbladder is going to secrete bile in there to help you digest and stabilize, make these bile acids so that you can transport cholesterol. These are things like taurocholic acid. They're going to emulsify these fats in here. They surround droplets of fat, increasing the surface area. So you basically have this big droplet of fat in there, and then lipases can start chewing them up. Okay. Other tissues will then work on converting the cholesterol into steroid hormones and so on. Okay. So if you look at our cholesterol, you can make hydroxysterols in there. Okay. You can make bile acids. So here would be an example of a bile acid, taurocholic acid. Here you have a cholesterol ester, where you take this hydroxyl group, add an R group on there. And then of course we can make our steroid hormones, like maybe somebody who's working on, a senior seminar paper about progesterone. Well, pregnenolone, here we go, as a precursor. All that good stuff in there too. Okay. I'm going to, let me look at what slide we're on here. Okay. Ooh, man, this is a long chapter. So just to introduce this, I'm going to look at this, but we only have a couple minutes left, so there's no way we're going to get through all this. But we've got another 24 slides, 26. Yeah. Do 10 slides a minute. But this whole next section is talking about cholesterol transport and lipid transport through the body. So lipids are carried through the plasma on spherical particles. Remember, they're hydrophobic, so they don't dissolve in the bloodstream. They need carrier transports. So what we see is, you can think of this as like an M&M. We got a crunchy shell and a creamy center in here, okay? Or maybe a Twinkie would work better. All of our squishy fat, or as my one professor that I still can't, our greasy parts of our lipids in there are on the inside, and our proteins are on the outside so that they can dissolve in the bloodstream. And of course, we'll have various proteins out here that can interact with receptors. So like our ApoB proteins, we're looking at apolipoproteins, all right? So these are surface proteins that deal with lipids, so apolipoproteins, and there's of course a phospholipid monolayer out there. The interior is just filled with fat, and some proteins are inside, but we'll see. The content and the size and the makeup of the surface proteins define about 5 or 6 different particles that we look at here. Okay, so we're going to classify these as 4 major particles. There is actually an IDL for intermediate in there, and I think there's another weird one in there. But these are the 4 major ones, and they're based on what we call sedimentation, but it's really density, and what's the makeup of these. So we have chylomicrons. Those are the fattest. If you think of Kylo Ren, it's like that's the weakest part of Star Wars, so full of fat and squishy. I don't know. Chylomicrons are the largest, They are also the least dense because they're mostly fat, and fats and oils tend to float in there. There's very little protein in a chylomicron. There is a fair number of phospholipids, but there's a huge number of triacylglycerols. We typically see these for transporting. Once we get out of the portal vein, we've eaten a fatty meal, chylomicrons are going to be transporting stuff in there. Our VLDLs are less dense than chylomicrons, sorry, more dense than chylomicrons, but still relatively low density. Very, they've got more protein, but not very much protein, 10% protein. A lot of phospholipids in there, some free cholesterol and some cholesterol esters. So we see our VLDLs as our, as a sort of transporting cholesterols in there, but we also have a lot of triacylglycerols. When it comes to cholesterol in the body, the main thing we look at are LDLs and HDLs for low density lipoprotein and high density lipoproteins. And we can see, our HDLs are mostly protein. Sorry, I should say vesicles. And our LDLs are about a quarter protein. There's a fair number of phospholipids in there. We can see LDL is our primary carrier for cholesterol. Okay, very few triacylglycerides in LDL. HDL has even less than that. HDL also has the second most cholesterol in it after our LDLs, and HDL is involved in scavenging cholesterol. So LDL transports it where it needs to go. HDL soaks up excess cholesterol. LDL is sometimes referred to as the bad cholesterol and HDL as the good cholesterol, but really it's probably more of a ratio in there. HDL is involved in clearance of cholesterol. LDL is involved in just transport. If we look at these, we can blow these up by size. This should actually be 3 times bigger for comparison. Chylomicrons are big and fluffy. They're the giant marshmallows that you find in the store. VLDLs are smaller, but they're still pretty fluffy. Our LDLs are small and yeah. These are mini marshmallows. These sprinkles. Yeah. We're in your Lucky Charms. Yeah, they're kind of like crunchy. That's terrible because you should not eat Lucky Charms. Yeah. It's like nothing but marshmallows. Okay. When we get back for spring break, we are going to go into detail. We're going to talk about some of these apolipoproteins. Do you need to memorize these? No. There will be a handful you will want to memorize, but not all of them. And yes, we'll go through that. Yep. Oops. Yeah. Well, 3 or 4, I think. But like binding the to the LDL receptor, you're going to want to know ApoB100. That's the one that's critical for binding to the LDL receptor. My favorite is ApoE because it clears VLDL and chylomicron remnants. So once we transport these, these bind to receptors and then kind of vomit out all the greasy fat particles into the cell, and then they're just kind of like shrunken like a deflated volleyball or something like that is left. So the ApoE triggers the clearance of those as well, because you don't want the remnants to stick around in the bloodstream because they can be involved in plaque formation as well too. But also like ApoE because last name starts with E. But you can see interesting things here like ApoC2 and ApoC3 that will activate or inhibit lipoprotein lipase. Probably don't need to memorize those. I think B100, our LCAT, which we'll see, this is going to be a transporter in there, but I know I'm going over time now. So we will pick up with this slide number 85. All right. It's a long freaking chapter. Yep. So I'll let you get ready for that. Let me stop sharing here and stop recording.